It's really been very, very nice. Is this on? Yeah. So I'm the last speaker, and I'll try to stay on time. I'm impressed that there's still people here on a sunny, warm day in Paris, but it seems like a pretty... Yeah, I doubt that. So, how do I... Okay. So everyone shows where they come from and their first slide, so I'll just show you where oh, wait, that was last night. Sorry. This is actually where I come from. This was a typical day from December through March in New England this year. It was pretty horrible winter, so that's pretty much all we saw all winter. I live next to a horse farm. I don't have horses, though. So this is the program that I built at the University of Connecticut Health and School of Medicine, including a team of gastroenterologists right here, Dr. Devers, all the samples that you'll see today. He's the one that did all the hard work to do the scoping, the endoscopies, the chromoendoscopy, and Joel Levine was the one who formed the program with me many years ago. And so there are a number of cancer risk factors, and I see why everybody stands back here, that are associated in the colon in the West. And of course, the lifetime risk is increased in men and women by probably around 65 is sort of the peak. There's about 160,000 cases a year, so it is the second leading overall cause of cancer-related deaths. There are a lot of risk factors, not just heritable FAP and HNPCC, but also a number of other risk factors primarily associated with diet. Diet is really, and also, believe it or not, tobacco is also a major risk factor for certain subsets of colorectal cancer. And I think diet is best reflected over here. This was actually a barbecue that I was at, and it was actually delicious meat. The guy who discovered leptin about 20 years ago, he has a big barbecue every year. He likes to eat a lot of red meat. So obesity, diabetes, refined carbohydrates, lack of exercise, and then the Western diet or the fast food syndrome. The French have been amazing in eating a lot of fat and meat and yet not having that much colon cancer, I think, until the arches of McDonald's showed up and probably sank everything. But we're actually collecting lots and lots of this type of epidemiological or nutritional epidemiological data. We've just codified about 2,500 consecutive colonoscopy subjects in terms of their risk factors. We have a paper we just submitted looking at a number of interactions between some very interesting and important both risk modifiers, suppressors as well as inducers of colon cancer risk. We know that there's decreased risk from using NSAIDs, and it's not just aspirin, but even ibuprofen is also protective. Prior endoscopy, obviously, calcium, vitamin D has some protective effect. There's a lot of incomplete data on folate, which is loaded in our Western diets because of women that are pregnant and we want to prevent neural tube defects. But the way our governments usually work is find something that seems beneficial to a subpopulation and then apply it to the entire population. And the problem with folate seems to be that if you're a 60-year-old male with a high-grade adenoma, it may actually be a really bad thing. And so my laboratory is actually working very hard right now on folate and methyl donors to see whether or not we can interrupt tumorigenesis by affecting methyl donor availability. Vitamin E has been very variable, and then resveratrol and curcumin have been largely disappointing in terms of prevention of cancer, of colon cancer. But certainly chemoprevention, which is an area of research that I work on a lot, suggests that targeting key risk factors could be the key towards preventing colon cancer. Also, probably not living in Texas. I was there about a month ago, and if you think that previous slide of the red meat was impressive, this is impressive. I mean, I don't know why everyone in Texas doesn't have a lot of adenomas, because that's a serious amount of burnt charcoal meat. And I don't know that they take NSAIDs and drink red wine as much as probably people in Europe do. So this is a well-established and old pathway of colon cancer. I actually stole it from a collaborator at the University of Utah a number of years ago, but it's a really nice pathway that shows you how you go from a normal colon to an initiated epithelium. This is the area that I study and I'll provide most of my data from. A number of initiating mutations like APC, KRAS, and BRAF drive a normal colon towards this initiated state, and then these other factors that I've talked about like obesity and inflammation drive one towards early late adenoma, and then later acquired mutations in the colon such as P53, TGF-beta, there's many others, can lead towards promotion, progression, and carcinoma. So I'm not a hardcore techno guy. I know there is a lot of technology theme in this talk. These are some of the instrumentations that we've used, and primarily a lot of the stuff that I'll talk about today. High-definition chromoendoscopy, laser capture, using way back in the Pixel 2E days when I started doing LCM in '99, Veritas, and then the XT platform. DNA sequencing, mass spectrometry using Sequino mass array. We've done some work with nanofluidic proteomic immunoassay. I'll show a tiny bit of data. And then RRBS, which is a way to look at the entire methylome, or about 85% of it, and I'll have some unpublished data at the very end. David Drew did a lot of the work that you're going to see today. He's finishing up his PhD within the next few months and moving up to Boston, unfortunately for me and fortunate for him. So this is what we study. This is a normal colon, a screening colonoscopy, and these are aberrant crypt foci. They're very, very small. We began by studying them in the distal 30 centimeters of the colorectum from the splenic flexure down. And in order to see them, one needs to spray the colon with a dye. In this case, it was methylene blue dye. Now we use indigo carmine, which is actually the dye that's found in blue jeans. And so, you end up having a colon that temporarily is the color of Levi's jeans, but it's not toxic. So this is actually a captured aberrant crypt foci. This is one that we've left inside. I have an ongoing study now that allows us to collect up to 12 of these biopsy specimens. They're up to 5 millimeters in size, so they're very, very small and typically can't be seen without this sort of high-definition endoscope combined with dye spraying. And for the first part of the talk, I'll only talk about the lesions and their characteristics in this part of the colon. Later, I'll move over towards the proximal colon. So are aberrant crypt foci, or ACF, cancer precursors? That's a big question that is raging, at least in my mind, if not in many other minds. So the traditional Vogelgram goes from a normal colonic mucosa through the dysplastic aberrant crypt foci, or a microadenoma, to a traditional adenoma and then to an adenocarcinoma. And again, involving some of the tumor suppressors and oncogenes that I talked about. So this is what's referred to as the traditional pathway. Jeremy Jass, about 10 years ago, with pathologists, late pathologists, identified an alternative pathway that's more active in the upper or proximal colon. And this is referred to now as the serrated pathway. And the serrated pathway has a little bit different temporal set of changes involving BRAF, KRAS, but also a lot of methylation, SIMP, as well as microsatellite instability. And this pathway goes through possibly hyperplastic ACF, which we find very common within the colon, both serrated and distended, to ultimately to a sessile serrated adenoma, typically in the upper colon. Probably 25 to 30% of colorectal cancers are going through this pathway. And even though there are not that many precursor lesions that look like this, probably 30% or 25%, even though they're very infrequent relative to the traditional adenoma, they have a much higher likelihood. They're much hotter lesions of progressing towards cancer. They're very, very interesting. They're often missed because they tend to grow in a very flat manner, so they're hard to see endoscopically. Whether or not the hyperplastic ACF can convert to dysplastic microadenomas, we don't know. There's just a brief highlighted history of the aberrant crypt foci, first described in 1987 by Bird and colleagues in Canada, and a number of studies over the years. In 1991, Teresa Pretlow described them, as well as Roncucci, in human colon. These were in resected colons from cancer. They would spray the colon on a bench and then look for these lesions. And then really a Japanese group, Takayama, published in New England Journal in 1998, that they could actually see these lesions in situ during a colonoscopy, and if they gave patients aspirin, they could actually see them regress, or there would be fewer of them if they went back over time. Finally, there was a large cancer prevention network trial by Paul Limburg at the Mayo Clinic that tested a number of different potential chemoprevention agents, but did not have a lot of success in looking for the numbers of ACF in people consuming different chemoprevention agents. There are a number of reasons for that, and I don't think that that story is really complete yet. So about 2 years ago, we published really the first paper that looked at a follow-up. This study looked 5 years prior to endoscopies that were done and ACF counted at our institution. And what we found in this one slide, I'm sorry it got sort of screwed up here, was that if you had more than 6 aberrant crypt foci 5 years ago, you had a much higher likelihood of having an advanced neoplasia. This got really messed up. But anyway, they seem to be predictive of a later acquisition of a high-grade adenoma. Everything that you'll see and everything we do is with microdissection because these lesions are so small. They're anywhere from 1 to 5 millimeters. And so everything that I'll show you will be separating hyperplastic or dysplastic crypts from adjacent or distant normal crypts. Just since there is a lot of technology, I threw an extra slide in. We've been trying to do fluorescence LCM. We've had a lot of struggles with it because when you do the immunofluorescence, it's difficult often to get high-quality RNA, but I think Lance is really is leading the way, and I feel very confident when I go back that we may be able to get some great data. This is just showing where we don't have to do immunofluorescence. This is actually from a GFP-labeled LGR5 mouse. So these are mice that have LGR5 lighting up the base of their intestinal crypts, because LGR5 is as you will probably all know, a stem cell marker in the small intestine and probably in the colon as well. So here we don't have to actually do any reactions. The mice do all the work for us. So we can simply laser capture fairly intact in fluorescent cells. By combining this with other mouse models that we have, we may be able to actually look at APC mutations and look at the cells at the precise time that they're losing the APC wild-type allele. But I'm not going to get into any of that work right now. This is some of the work that we've published over the last number of years using laser capture microdissection on these aberrant crypt foci. So we published a paper a number of years ago looking at hypermethylation of a putative tumor suppressor gene called RASF1A, when we found and confirmed by PCR that RASF1A was actually being hypermethylated in about a third of these lesions. We identified an APC mutation, actually a novel APC mutation, in a cancer research paper a few years ago, confirming the mutation by looking at beta-catenin immunofluorescence, and you can see some nuclear beta-catenin here. The only time we've ever seen the APC mutations are in these lesions that have already acquired dysplasia. We've also, I'll show you data on the nanofluidics proteomic approach that we've used. We published this last year looking at ERK signaling in these lesions. And I'll show you more data on that. We also found microsatellite instability, and this was published in Carcinogenesis a few years ago. So there are a lot of molecular genetic events that are going on in these lesions. Despite their small size, they do have an awful lot of interesting biology or cancer biology within them. And along that, this is a workflow where we look at RAS mutations and BRAF mutations in normal mucosa and then in two different types of hyperplastic aberrant crypt foci. One we refer to as distended because they're morphometrically expanded large crypts. They're not normal crypts, but they're not dysplastic at all. The other are the serrated aberrant crypt foci, which are simply crowded crypts that have actually started to form involutions and folding because there's probably just too much proliferation and too little death. And so we microdissect, IR microdissect, and then extract either protein or lysates, which you'll see shortly, or DNA. So here we looked at a spectrum of KRAS mutations, and you can see the frequency in both distended and serrated ACF. The G12D was the highest hit that we would typically see, and that was fairly equivalent across both types of histologies. What was interesting to us, and the reason that we started looking at proteomic changes to ERK, was that BRAF seemed to be almost always confined to a serrated histology. Rarely, if ever, did we see distended ACF with a BRAF mutation, which was very interesting to us because the BRAF and KRAS obviously share part of the same pathway, and yet they have a different phenotype within the colon, within the human colon. So one question I have, and I don't have the answer at all, is whether there is different oncogenic potency of distinct KRAS mutations. These questions have been asked over the years. Really, to answer it in a very, very careful way would be very, very difficult. Probably would require mouse models and some fairly elegant work to actually understand whether or not these different oncogenic mutations may ultimately have a different effect in the human colon. But I don't have an answer for this. So the phosphorylation signaling events that I just mentioned through RAS and RAF, we're going to focus on phospho-ERK1 and 2, leading typically towards proliferation and activation of MYC, many other changes. Wow, that did get screwed. It's still the Mac PC war going on. It's amazing. So KRAS is mutated commonly in colorectal cancer. BRAF is also typically, you've heard about the V600E substitution mutation. About 10% of colorectal cancer. Mutations to BRAF and KRAS are mutually exclusive. You never see them in the same lesion. And ERK1 and 2 carrying dual phosphorylation, so ERK1 and 2 both have two phosphorylation sites at their tyrosine 3-enine residues, go to the nucleus where they drive mitosis. So we're going to look at ERK phosphorylation status, single and double ERK1 and ERK2, in aberrant crypt foci that have sustained a RAS or a RAF mutation. And first we tried to do it using immunofluorescence, and this was published in Proteomics last year, where the red is ERK1 and 2, single antibody, and then the merged image would be nuclear ERK1 and 2. And what we found here by immunofluorescence was that there was a nuclear accumulation of 1 and 2 suggesting activation, but it's really difficult to quantify this, especially with a single antibody approach. So I was at an AACR conference several years ago, and I saw this relatively new technology called a NanoPro, which was a method for doing sort of a virtual Western blot on a nanofluidic tube. And so we decided to, they had some very nice data on ERK phosphorylation, so we decided to develop a protocol to collect the ACF biopsies, and we get them, many of them every week. We get several patients, up to 12 biopsies, prepare them in frozen OCT, and then do protein extraction and isolate lysates and run them on this nanofluidic method. And this is the NanoPro that we used. The methodology is here. It's all been published a number of years ago. But you can only really look at a single antibody, and it's got to be the perfect antibody. As it turns out, it's a very, to look at more than ERK at this point is very difficult. It's a very challenging method to develop and to develop new antibodies against specific antigens. So we've really been limited to looking at ERK. And so the next set of data, I'll show you what happens to ERK1 and 2 using this nanofluidic method in these lesions. And the readout looks like this. ERK1 and 2 are perfect in the human because they all separate clearly on this chromatogram. We've actually tried doing ERK1 and 2 in mouse, and there's overlap of two of the peaks, and it's been very, very challenging just going to the mouse from the human. But the bottom line in this published work, which I won't belabor, is that we see both primarily ERK2, very strong ERK2 activation in aberrant crypt foci that have a BRAF mutation or a KRAS mutation, but we also see it in wild-type ACF. So the conclusion we made from this is that something activates ERK, and it doesn't have to be BRAF, it doesn't have to be KRAS, probably EGFR signaling is just switched on from a number of other mechanisms, but ACF certainly have activated ERK signaling. One thing we did notice was that phospho-ERK1 and singly and doubly phosphorylated ERK1 were not activated in the BRAF group. And there is some evidence that doubly phosphorylated ERK1 may actually competitively inhibit ERK2. And so perhaps there's a stronger response, maybe, as far as proliferation goes in the ACF because of BRAF possibly inhibiting or not activating the ERK1. But we don't know any of this. Now, for the last part of the talk, I'm going to focus on proximal or right-sided aberrant crypt foci. So these are lesions that form beyond the hepatic flexure. These are the upper part of the colon, sort of the last frontier of the colon, and maybe the most interesting part of the colon because this is where these interval colon cancers form. And what I mean by that is you go in, you have your colonoscopy when you're 50 years old, and everything's fine, and then 3 years later you find out you have metastatic colon cancer. And often it's because of lesions that are forming and being missed in the proximal colon. Whether that's developing in that small interval of time because of different growth characteristics of the lesions or simply missed endoscopy, everything may contribute to it. But there's no question that this part of the colon is a very hot area to study in terms of premalignant lesions converting to neoplasia. So this is what it looks like. This is the indigo carmine. It's different than the methylene blue. It's more of a greenish hue. And here's a flat adenoma that's forming. They're very, very hard to see. And here's an aberrant crypt foci, actually, in the proximal colon. And this is Dr. Devers' forceps that he'll take out. Pretty much he'll take out all of the proximal lesions. And the reason is that the counting-wise, they're very infrequent in the proximal colon, the aberrant crypt foci. They're very common in the distal colon. In fact, we've seen as many as 100 distal colon aberrant crypt foci, but in the proximal, the average is 1 or less. So everyone has multiple aberrant crypt foci in their distal colon, but not that many here. And we actually think that these lesions in the proximal colon, obviously they're self-limiting, because if they weren't, everybody would have colon cancer. So we think that the oncogenic mutations, which I'll talk about in a few minutes, are actually activating some sort of senescence pathways that are protecting your colon from going on towards malignancy. We think in the proximal colon, some of those pathways may be evaded because of hypermethylation. So how much more time do I have? I lost track. 5 minutes. Okay. So this is our data set. This is our sample population right now. What's that? Yeah. Okay. We've done about 200 subjects so far and collected an awful lot of demographic information on them. We've also, as I mentioned before, done 2,500 consecutive colonoscopies looking at risk factors in adenomas, but those 2,500 weren't dye sprayed, nor were they fresh biopsy. But all of these subjects have been fresh biopsies, up to 12 lesions per colon, blood, food frequency questionnaire, and demographic information. So the histomorphology, the frequencies are quite different. We have the hyperplastic ACF, the enlarged and the serrated, then dysplastic ACF, and the dysplasias tend to form in the proximal colon. About 57% of those proximal lesions tend to have dysplasia, and we're beginning to just think that they're just simply microadenomas, very small lesions, and we don't see nearly as many of the hyperplasias in the proximal colon. I'll skip that. So we decided, based on some of these characteristics of right-sided ACF, to expand our genetic characterization to try to figure out what these proximal right-sided ACF mean, or what's going on in them. So we use DNA mass spectrometry using a sequenome mass array system, which is a commercial apparatus that sort of, I think it's kind of dying out. We were doing the work at Yale, and the service contract was so expensive, Yale dropped the instrument, and we're having trouble finding this instrument to use. I don't know if you guys probably know. So,
no, but yeah, it is dying out.
But it's a very nice approach, but unfortunately, it's just, I guess, too expensive to operate. So this work that we've done on proximal ACF will be coming out in 2 weeks in Molecular Cancer Research, and we're pretty excited about this study. It was a pilot study, and we're following with a much larger study. The mutations that we screened, we did using a off-the-shelf OncoCarta commercial panel, the OncoCarta version 3. These are some of the tumor cancer-related genes that were in the OncoCarta panel, and some of the specific mutations that were screened are shown here. There were about 120 mutations altogether. The problem with this panel was that it wasn't designed for a colon. It's a sort of a general cancer panel. And the readout for the sequenome is sort of a typical chromatogram where you look at the area under the curve to make calls. And this is just giving two examples. We found an EGFR mutation and a FLT3 mutation, neither of which had ever been shown before in colon cancer. According to Cosmic Database, EGFR alterations are in approximately 6% of colorectal cancer, FLT3 and maybe 5%. So I think we're on the right track here. And again, this work will come out in several weeks. We also found a new APC mutation in a proximal dysplastic ACF, which is shown here. We know it's a somatic mutation, an acquired mutation, because we didn't find it in 3 synchronous adenomas, nor did we find it in blood. So we took out this tiny little lesion from the proximal colon of a normal subject, and may have, who knows, but maybe we prevented a subsequent neoplastic or serious neoplastic growth down the line. So I think that was good. We confirmed the absence of the mutation in tubular adenomas and blood, and we also looked at, confirmed functionally that it had a functional consequence by looking at beta-catenin and found it was nuclear. So in the last couple of minutes, I'll just show you our follow-up study, which is we customized the sequenome panel and focusing more on some of the genes that we really wanted to look at. And what we found was quite surprising. These are all mutational hits that are positive, called positive by the sequenome analysis of 20% ratio or more. So APC, BRAF, EGFR, HRAS, KRAS. So what we're surprised by is the likely mutations. And here we found, and these are probably, most of them going to turn out to be correct. We're going back and at least validating all these mutational hits. But what's surprising is that there were 8 HRAS and 11 NRAS mutations. And I don't know that those 2 mutations are not common in colon. So I'm not sure why they're there. I'm not sure what they mean, but we're trying to figure it out. And that's sort of a story that hopefully we'll have soon. This is just the primary hits that we found. This is just showing you that in the large intestine from the Cosmic version 53, actually, in the large intestine, less than 1% are HRAS mutations and less than 3% are NRAS. Most are KRAS mutations. So we are surprised by this result. I'll skip the summary so I can get to the last set of data. And I'm going to skip. We're doing a lot of single cell now in the colon, but I'm sorry. So we're interested in oncogene-induced senescence as a mechanism to protect the colon from oncogenic mutations that I've described, like BRAF and KRAS or EGFR. Oncogene-induced senescence is a mechanism that enables cells to avoid becoming neoplastic by simply going to a senescent state. It's a very complex set of mechanisms to lead to senescence. We just are looking at the simplest one, which would be P53. So in the left side, we hypothesize that RAS-RAF activation causes hyperproliferation, P16 goes up, suppresses hyperproliferation, and we go into oncogene-induced senescence. But in the proximal colon, where there are very few lesions, we predict that RAS-RAF activate hyperproliferation, but what happens is that there's concurrent hypermethylation of important tumor suppressor genes like P16, which block the oncogene-induced senescence, enabling these lesions to have a higher likelihood to progress. We've looked at P16 by immunofluorescence, and it's very difficult to quantify. It's almost impossible to quantify, to make any distinction between a KRAS or a BRAF mutated aberrant crypt foci and the amount of P16 activation. So Matt Hanley now, in the last two slides, is a graduate student in the lab who's quite interested in epigenetics. And in collaboration with Gerd Pfeiffer, who's at the City of Hope, we've worked together in the past. Gerd uses something called RRBS, or reduced representational bisulfite sequencing, which is sort of a global epigenomic way to look at CPG islands, about 85% of CPG islands, and it's much less expensive and much more cost-effective than whole genome bisulfite sequencing. And the methodology is here. It's all published. But in our first look in unpublished data, Matt put together a volcano plot of our first set of data that we got about 2 weeks ago. And this is looking at aberrant crypt foci versus normal and looking at a percent change in methylation. So on the right side are hypermethylated CPG islands. On the left side are hypomethylated CPG islands. Again, comparing ACF to normal. And we're stunned by the amount, the extent of hypermethylation that's going on. We haven't begun to sort this out in terms of location in colon. It's on 20 samples so far, all laser capture, microdissected. Again, that's how we did all this. So this is really the first use of LCM to go into an epigenomic type approach to look at everything going on in the human colon. We looked at a number at six different methylation markers that are part of what's called a SIMP panel, which is used to determine the extent of hypermethylation in tissue. And we're finding that the hypermethylation of P16 is pretty much in every sample that we've looked at, which supports our oncogene-induced senescence pathway. But again, we haven't done any of this in terms of breaking this down in terms of location or histological changes yet. And epidemiology, we have all of that data. So I'll end there, thanking the NIH for providing a lot of funding, state of Connecticut tobacco funding money, stem cell money, diet initiative, and then the AICR for funding some of our research as well. And I'll stop there. Thank you.
If you take this plastic foci and you transplant them into xenograft, will they grow? Or if you treat them with growth factors, appropriate growth factors, will they grow?
It's a great question. I think probably 10 years ago we tried to do it. Before we really knew what we were doing, we haven't tried it. They didn't grow. They lasted for a day or 2 days, and we looked, and everything was dead. So I think now, if we can, establish organoids out of them and put them in, they will grow. And that's what we'd like to do.
So my next question is about BRAF. So if you have BRAF mutations, you might think a BRAF inhibitor would do something to these lesions, but I don't think you can give a BRAF inhibitor in this situation. However, a melanoma patient who is getting a BRAF inhibitor, we know most melanoma patients get BRAF inhibitors, may probably have had a colonoscopy also. So you could look at a melanoma patient who had a colonoscopy and then got their BRAF inhibitor, and then you could look at another colonoscopy and you could see whether there's regression of these adenomas by the therapy that the melanoma patient's going to get. And it would not, I think that would be accepted by an IRB, and you could specifically look at your hypothesis that way. Same for an EGF inhibitor that patients get as well.
That's a great idea. I mean, the problem with chemoprevention is you want to use very natural or very non-toxic agents in order to prevent cancer in the future. And you're absolutely right, but we could exploit trials or going on and see what's happening. It's a good idea. You guys want to get out of here?
Yeah.
So you showed that you see KRAS mutations in your ACFs in about 50%, in colorectal cancer 30%. In the adenomas you find these HRAS and NRAS mutations, which are rare in colorectal cancer. So what is your feeling about the adenoma carcinoma sequence then? Is it really...
I'm not saying anything about Robert Weinberg. If we can confirm those N and HRAS, and they have strong oncogenic activating activity, they might just be really forcing those aberrant crypt foci into a quick senescent pathway. But I don't know that we disprove in any way the Vogelgram, other than it's just probably a little bit more confusing than anybody would have thought. But it's a perfect, valid question. So I don't have an answer for that yet.
When you look at multiple lesions in the same patient, are they similar in any way? Or are they, it looks like they're all coming from the same like primordial change or germline predilection, or are they completely different in every patient in every lesion all over the place?
That's the problem. In the distal colon, you can see a KRAS, you can see an APC, you can see a BRAF. So you think there's a field effect going on, and there may be, but it's not confined to some primordial change, which really complicates it. It makes it difficult. So.
Here. So you think there is oncogene-induced senescence, but at the same time, senescence can be, there is a secretory phenotype that can induce cancer as well. So what do you think about it?
It's an excellent point. I don't know how to answer it. I mean, we're thinking it the most simplistic way, that the senescence is protecting and that it's preventing cancer, and the lesions are disappearing simply based on their frequency and the low frequency of cancer. So that's our dumb explanation, is that it's just activating that one type of senescent pathway. But I'd be open to any suggestion at all.
I have another question. In these ACFs, have you looking at for genome instability? Because telomere shortening is quite often, right?
Yes. We did CGH arrays about 6 or 7 years ago using SNP chips, and they were very stable. It was so disappointing. I mean, we invested a lot of time and money in it, and really they didn't look very interesting. But again, we never published any of that data, and if someone who's used to looking at that data, much more expert than I am, they might be able to find something. I know that Thomas Reed at NCI has argued that trisomy 7 is he argues a very early event in colorectal cancer, very early. We didn't really see evidence of it. He's published that, but I'm not sure. So there may be subsets that are going through some sort of genomic instability very early, but at this point I can't answer it. But thank you.
So you were saying that BRAF and KRAS mutations are mutually exclusive, as NRAS and BRAF are in melanoma, and but you were showing the mutations in HRAS and NRAS also. Are they also mutually exclusive with the with the BRAF?
That I don't. We haven't yet begun. That's pretty new data.
Okay.
So I'm not sure yet. So I think they are mutually exclusive. I don't think David found multiple mutations of any of the RAS family members at the same time in the same lesion, but I'm not 100% certain.
But then the question, I'm not very familiar with these mutations in colon cancer. Are they expressed then, NRAS and HRAS, regularly?
I'm not sure. I mean, RAS, of course, it's strongly expressed under most conditions in the colon, but NRAS and HRAS, I don't know. I'm not sure. We have to go. I have to figure that out.
Because what they say is that they are localized, differentially localized in the cell, so they are not exactly all. For example, KRAS is more in the lipid rafts, I think, and the other ones are so. There are different localization in the cell, and then maybe this could help.
If we can validate it, I think there's an enormous amount of cell biology that could be done to look at these questions if we validate it. Again, they were in the less than 20% hit rate, so we're not 100% certain that those are true mutations yet. So that's why we haven't published any of that data yet. Thank you.